people are back. So thanks everyone. Hopefully you had some fruitful discussions. So I just thought we'd just spend the last sort of 10 minutes sort of reporting back on the main, like a synopsis of what we've been discussed. So I don't know if Zahir, you'd like to start with your group, please.
All right, I can. I think I'll be quick and brief because we will produce this report later on, but we had a very sort of a lively discussion there. In terms of the question number 1 for the knowledge gaps and methodologies, there was a sort of a big consensus that although we have now emerging methodologies, the question is, are we still measuring the right things? Because the question is that how best we can link exposure to health outcome. That only sort of you can answer it if we start looking into the thing at the beginning in terms of that looking at the health sort of indicator endpoint, and that indicator endpoint will vary according to the environmental scenarios. For occupational, you might need to be looking at something completely different when you come back to the environmental scenarios. So really need to sort of start looking into developing those kind of indicator candidates of exposure. For example, the question was this, that endotoxin is still good to look into indoor exposure to fungal, let's say, exposure from damp buildings. And there was another sort of a question we better need now to look into the role of human microbiome, and then it's a relation to the exposure, and that leads to the inflammation, for example. And also start looking at sort of genetic predisposition, or in terms of the co-exposure or comorbidity scenarios where people are the sort of fraction of population if they have something else already. So how they are sort of exposed to biological agents and in terms of public health outcome. And for the second one, I think the nutshell is this in 2 ways. For the second question is that that it is very difficult to understand sort of complex exposure because of the complexity in the biological agents to relate it back to understand the harm of mechanisms, to understand the mechanism of harm basically. Now, I think one of the sort of comment was this, that perhaps that we need to better develop our cell models, and also we go back to some sort of animal model system to understand this complexity of multiple exposures from different biological agents that we are exposed to come up with, let's say, to better understand this mechanism of harm. And that can be only achieved by developing, let's say, a better sort of cell model systems. Thanks.
Thanks, Zahir. Fred, do you want to go next, please?
Yeah, we get cut. We were starting to have a lively debate. It's good. There were quite a lot in the room, people around real-time detection and quantification, and I think that it all agree there is a need for real-time monitoring on pollen, spore, and need to make a distinction also within the bioaerosol, what is more for allergen and what maybe more pathogen. So how we could do that, and revisiting also data. Some of them are quite old, and it was also mentioned about validation, so trying to understand these dose exposure. There is the seasonality that's been mentioned, and also trying to understand also with age group, perhaps also within the life cycle of a human, that it might be different exposure might trigger later on different response or allergy. So this is not well yet understood. And we were looking also, there is also perhaps interest on linking also with climate change and what might impact this exposure. And then we were starting to consider and discussing, but we haven't time to continue, but effectively within the bioaerosol is one aspect is more about triggering or responsible for allergies, spore and pollens, but also what might trigger immune response, perhaps with more pathogens aspects.
Great, thank you. So I can we talked about similar things as well, but one of the things I guess similar with the first group is the difficulty in getting that good exposure data and population data versus sort of personal exposure data, and what's required and when it's required and how it's required. Andre sort of commented on his ideal data set, which would be you'd have a long-term exposure data for as many pollutants as possible. And then you'd match that with some health data, which is for the same sort of period or even longer. And obviously that would be ideal for everybody, but that often doesn't occur in practice. And so it's making the best of what you have and thinking about what we could develop, and could we develop some data sets that are available to more people. And that got us thinking about data sharing and how it can be difficult because sampling and that is so labour and resource intensive. It sort of discourages sometimes some sharing. So it's thinking about bringing experts together to kind of prevent that silo working and getting people to talk more about their area of expertise and sort of common interests and where we can use them to sort of break down some barriers. We also talked about, in terms of new technologies, what sort of real-time monitoring there might be out there, like the second group. Identifying potential proxy methods, like Anna talked about, biomarkers or other proxies, like distance maybe is a very simple one for measuring things and how we can improve that. So thinking about what kind of modelling might be useful in that respect. And then with question 2, again, we didn't really have experts within sort of the modelling side or the sort of cell model side, but we were thinking about, you could have experiments to establish different levels of exposure in different parts of the body and therefore sort of what impact you might have on those systems. You're thinking about there's a trade-off between doing a whole systems approaches versus looking at specific pathways and maybe having a battery of in vitro type sort of experiments that you could use. And engaging as well with practitioners, so asking the questions of them and what they're interested in. So I think we've covered quite a lot of different areas, and I think we've highlighted quite a lot of different issues and areas that need focus on. So I think really this network, one of the aims of it is to get to like Simon mentioned earlier, is to break down those silos and get the different experts talking. So hopefully today we've given you kind of an overview of what this theme is about. Obviously there's overlaps with the other themes, the other 3 themes within the network. So I'd encourage you to get involved with as many as you would like. And we really want to think about this sort of holistic approach, sharing data, sharing expertise, and thinking about different things that input into this very interesting area. So if you have any ideas on future sort of focus for these for some more events in theme 3, would you like something on modelling? Would you like something on cell models, animal models? Please get in touch. And if you've got any other colleagues that would be interested, or you could suggest people that could input and provide presentations. We've also started to think about whether, in order to get people talking, some sort of Cam suggested some briefing papers from different experts to help each other understand each other's expertise might be useful as well. So please, if you've got any other ideas, then just get in contact with one of us, and you can do that via the bioaerosols webpage. So we're at 16:58, so I think everybody's had enough of Zoom. I'd like to thank the speakers again for their excellent talks and for everybody for contributing. So thank you very much, and we hope to see you soon talking about bioaerosols. Thank you.